Okay, guys, let's finish up with this chapter 1, kind of chapter 2 stuff. And this is part 3 of that chapter. And then we can start jumping into, start talking about some organisms, right? Get into some chordate phylogeny or some chordate taxonomy. So we were talking about how biologists like to try to determine common ancestry or evolutionary relationships between organisms. We're going to stick with vertebrates. And one of the both classical and modern ways to do this now is called cladistics. And it's a method that applies the scientific method to the construction of these phylogenetic trees. Remember, every phylogenetic tree is a hypothesis, right? And basically there are now, because of the advent of these supercomputers, the ability to use algorithms and really, really, really large data sets to try to figure out and group organisms based on ancestry. And we use what's called parsimony to build these trees, which is the least number of steps, all right, that it takes to get our tree made and to link organisms based on common ancestry and some of the common data points. We also use what's called Bayesian statistics, which allows you to do things like bootstrapping and a whole bunch of comparisons between the different branches to determine how likely what we're seeing is real. And that is a really, really complicated, again, supercomputer data analysis program that allows you to analyze your nodes, your branch points. So data sets can be anything really that you can get a lot of repeating information about or a lot of information about. So data sets can be DNA and RNA sequences and the differences between them, amino acid sequences, morphological characters, or even behavioral characters. So I've used, for example, sperm ultrastructure, all of these little structures and differences and nuances that you can find on mature sperm under the electron microscope to build phylogenetic trees, or start to build phylogenetic trees, if you will, on the relationships between lizards and snakes, for example, since I'm a herpetologist. DNA in the advent of the molecular biology, these have been kind of the stable or most universal way, most accepted way that individuals or people are now building phylogenetic trees. So you collect blood samples or tissue samples and you get the DNA sequences and you compare those DNA sequences among organisms. How similar or different they are determines how related they are and whether they belong within a evolutionary line. So part of this is determining what is ancestral and what we consider derived, right? Or something that has changed from the ancestral condition. And so cladistics is based on these kind of characteristics. Shared derived characters that make these organisms grouped together different than the ancestor are called apomorphies. Apomorphy means derived characters. Syn means shared. Similar is not enough. It requires some reflection of descent, right? So we can group animals, we already talked, based on they look alike, but we need some way to determine descent. That is, what is the common ancestor? And that requires characters that are called plesiomorphic, right, which I'll show you in the next slide, or characters that determine the ancestral state. And those are provided by what we call outgroups. So here are some characters that you could collect. Things like eye color, mammary glands, number of legs, length of bones, different architectural points on skulls, nucleotide bases, right? Whether there's an A, C, T, or G in a particular spot. Amino acid codons and how similar or different they are. These are all characters we can use to group organisms based on which characters they have. The big problem is finding out which ones are derived and which ones are not, right? Which ones are ancestral. Right, and sometimes that's easy to do, and sometimes it's not easy to do, and makes building trees really difficult. So we compare traits that are shared or not shared. Shared traits that are shared with primitive characters or with primitive organisms are called symplesiomorphies. Shared primitive characters suggest a distant or common ancestor. And as I mentioned, this is why we're going to use what we call outgroups in a minute. All right, an outgroup is going to basically root our tree, is what I like to call it, root our tree, all right, our phylogenetic tree. Outgroups are groups that have some of the plesiomorphies, but not all of them. All right, and so you would think that if I have some and not others, that there's some ancestors or there's some other intermediate groups between those two, right? And our outgroup at least gives us some information about what we would consider ancestral. So these shared primitive characters suggest a distant ancestor, and we can use these plesio. Plesio means old school, morphe means character or ancestral characters, that we call them synapomorphies and symplesiomorphies. All right, sim means same and syn means same. So symplesiomorphies are groups or suites of characters that determine the ancestral state and are often found at least partly in the outgroup. Not shared, but shared derived characters that are different than the primitive are called synapomorphies. So the way I think about it, to just simplify it for this class, is that these plesiomorphies define our clade. They define our group. You are a reptile because you have scales, right? And certain other characteristics, right? How do I then, if I get all the reptiles into a room, then how do I group them? I group them based on synapomorphies. Turtles have a certain suite of characters. Lizards have a certain suite of synapomorphies. Snakes have a certain suite of synapomorphies. Crocodilians have a certain suite of synapomorphies. Then I can, put them into smaller groups, and then take those smaller groups and put them into smaller groups, and your branches get more and more close to what we consider the true tree, if you will, or the true lineage, ancestral lineage of the extant reptiles. So shared derived characters indicate a more recent common ancestor. Plesiomorphies are obviously older, right? Characteristics that are shared by the entire group. And every once in a while, you get a character that's not found in any of the other groups that you're looking at. And that's called an autapomorphy. Autapomorphies are unique to a single species or a single group. Like an autapomorphy for turtles is the shell, right? There are no other organisms that have shells, extant reptiles that have shells. And so that's an autapomorphy for turtles. All right, so on our trees, the branch pointed where two species differed in a shared derived character, right? So there's a shared derived character that one has and one doesn't, for example. And in the in group, if you're in the in group, you're within the monophyletic group, the group of interest. You have the characters, all the characters in that group of interest. If you're an out group, your species or group that is closely related, you might have some of the characters of the in group, but not all of them. And again, out groups are really important because they root the in groups or root the tree. All traits shared by an outgroup, and the in-group must have arisen in a common ancestor that predates the divergence of the two. And you utilize those outgroups to really figure out what's ancestral within your grouping, or what I'm calling a clade. A clade is a true evolutionary group of organisms. So outgroup analysis is really important. If you can determine the outgroup, or we agree on the outgroup, then it helps us root our tree and figure out what's primitive and versus what's derived. So the lancelet here, or what we're going to call Amphioxus, is the outgroup of chordates because it has some of the characters of chordates, but not all of them. It develops kind of similarly. It has some similarities in the fossil record, but it doesn't have every single character of a chordate. So it's a nice outgroup that roots our tree. All other taxa and the vertebrates are grouped according to shared derived characters. And there are lots of vertebrates, right? Lots of chordates. So again, this lists our terminology. Polarity is simply we can distinguish an ancestor, which we say has the 0 for the synapomorphy, from the derived, which you give a 1, right? You give them 0, 1s, 2s, 3s, 4s, depending on how many characters you have. And that's what you throw into the computer, a code that it can then put together to see which groups are most closely related to each other. And your polarity is assessed, or polarity happens when you have an outgroup comparison. So you can see in this little simple phylogenetic tree, blue and square are plesiomorphies, right? So you can see here square and then blue color are obviously not found in the two derived groups. All right. Small size is an apomorphy for A, right? Or in this instance, could even be an autapomorphy for A, right? Red is a synapomorphy for A and B, right? They share it. circle is a synapomorphy for A, B, and C, right? So it's a synapomorphy. These are all circles. Obviously, this one is square. Blue is also considered ancestral because these two have red, right? So that's kind of how you look at a tree and determine apo versus plesiomorphy. Notice we use plesiomorphic here because they're not shared by the two, what we call ancestral groups, except for blue. Blue would be a plesiomorphy, whereas square is not shared with the circle, so it's a plesiomorphy by itself, right? So, building a cladogram. A cladogram is what you build when you do cladistics. All species are placed on the tips of phylogenetic trees, not at branch points. Each cladogram branch should have a list of one or more of the shared derived characters that are common to the species above the branch. Your plesiomorphic characters will be defined by your outgroup, right? And these characters are found at nodes, right, where the two branch points come off. All shared derived characters only appear once in a cladogram. If they appear more than once, then they're examples of convergence. And if you have a group that's really, really specialized that has lots of convergence, it makes building a tree almost impossible, right? You want to choose the most likely cladogram. The most likely cladogram are the ones with the fewest possibilities, and that's called parsimony. So how do we decide the best phylogenies? Parsimony, the simplest explanation, right? So here is an example of some fish here, requires only 4 steps. I mean, you can make trees that require all kinds of steps, right? This one requires 5 different steps. So the one with 4 is the most parsimonious, is the one that we would publish. So the fewest steps, the best. Remember, homeoplazy and convergence can really mess up phylogenies because it'll group organisms that look similar to each other that don't share common ancestry. So the convergence is really important, where you get the environment really shaping two groups of organisms that don't share common ancestry. And often the structures that look alike internally don't, right? But convergence can be a real problem in a lot of phylogenetic situations. And it makes certain groups of animals really hard to build trees for. So here's your examples here. So these are actually fish. So a lot of these are called cichlids, right? And cichlids are found in Africa. And we're looking at just this part of the cichlid tree. Cichlids are found in other places, but these are African cichlids here. And what you can see is there is a lot of species within the two lakes that look very similar to one another. And these lakes are separated from one another and been separated for a long period of time. So the common ancestors of these extant fish, they don't share a common ancestor. And this from on the left and right, right? Even though they often look alike, it's because the environment of these two lakes is kind of similar in as far as color goes, right? And shaping color. And that's how the species kind of differentiate themselves from one another. And you can see this is the true monophyletic group of these cichlids, even though you got a lot of overlap in morphology and color pattern between the two lakes. All right, so you got to be careful of homoplasies because they really, really can look like homologies and mess up your phylogenetic trees if you don't know where your samples were collected from. So classification and cladistics. You can see why we call true evolutionary groupings of animals clades off of cladistics. There are 3 kind of branching patterns that you can see. The one that we're trying to get and publish and figure out are monophyletic groups. Monophyletic groups are groups of organisms you can trace back to a suite of characters, a common ancestor. Paraphyletic and polyphyletic happen when we either forget groups or we combine convergence. So a taxon is any group of animal, right? It can be a genus, a family, an order. It is usually defined, or as best as we can, define them as monophyletic groups. But there are a lot of groupings of animals that we can't. You know, reptilia, for example, I study reptiles. Reptiles is not a real group because you either forget birds or don't study birds, or you link animals that we consider reptiles together to teach about them or to study them, and they really don't share common ancestry. So there are taxon that are unnatural, and then there are taxon that are phylogenetic groups that we try to keep together so that we can understand their evolutionary adaptation and their evolutionary history. There are also rooted and unrooted trees. If you don't have common ancestry, you don't have an outgroup, you have an unrooted tree. And the tree actually looks like a circle. And you'll have branch points that go out in a radius because there's no origin, right? Where if you get a rooted tree, it looks like this, right? This is your root. It just depends on the group and whether you can find common ancestors or not. So let's look at the differences between monophyletic, paraphyletic, and polyphyletic. It's kind of hard to see here, but I'll circle them for you, right? So over here, you can see this is what we consider a monophyletic group. If I'm just interested in L, M, N, and O, these are the living species, and I say they're a clade, you can see they're actually a true clade because you can trace them back to a common ancestor, C, right? If I did paraphyly on accident, and I studied all of these guys except for O, I left O out, then that would be paraphyletic, leaving a group out that belongs in the group. Good example of paraphyly is, I use this all the time, is I'm a herpetologist, I study reptiles, but I don't use birds. I don't study birds at all. Birds are reptiles, so I'm leaving the birds out of my group when I study them. I do that out of, not negligence, but just out of, it's easier for me because I'm more interested in just studying lizards, snakes, and crocodilians and turtles, for example. That's paraphyly. If we do it on purpose with the phylogenetic tree without explaining it, it's not proper, right, evolutionarily. Paraphyly is when I say, and I'll circle this like this, when I'm saying F actually has a common ancestor that we call B. It shares B with all the rest of these, where in reality, it shares C, doesn't it? So paraphyly happens when you add a group to a phylogenetic group that you think belongs there because of similarity. So often doing this happens in convergence, right? We see an organism that looks like a particular organism within our phylogenetic tree. I mean, they might have a suite of characters that look very similar to it, but we have to make sure that we use some technique that rules out convergence, right? And often what helps us rule out convergence is studying the DNA, right? Because DNA changes don't always necessarily revolve around phylogenetics, right? They don't, or I should say, DNA does not necessarily represent the phenotype that you see. Often the environment can shape the phenotype and the genetics can be pretty similar or pretty different. If the genetics are really different and you still get the same phenotype, that probably tells you that they don't share a common ancestor. So often with these cladistic trees, you want to do more than just DNA by itself or more than just morphology by itself. You want to do both DNA and morphology together, overlap your trees, see where they differ, see where they're similar. And often that can help you determine where convergence is a problem. Or you might get some information later on that shows F is more related to C than B, and then you would take F out, right, and fix your phylogenetic tree. Remember, each one of these is a hypothesis. It can be, you can get information later on that kind of disproves or does not support the original hypothesis. And this happens often and causes regroupings within animals. So that's the difference between poly means more than one ancestor. Para means you're leaving a group out. And mono means you got one solid group. And remember, we're always trying to the best of our ability with the information that we have today to find monophyletic groups. Okay, here's good examples of monophyly. So here, these are basically starfish here, what we call sea stars, right? They're echinodermatids. Echinodermatids, if you remember, echinoderma are a group that is sister to chordates, right? Or the most closely related group to chordates. We like to use them sometimes as an outgroup. And this shows you a couple of different what we call superfamilies. It's DEA, and they show your organisms in red, green, purple, and orange. And I don't know what color to call this down. Oh, it's the other red group, I guess. Well, it's kind of a maroon group, maybe. So these are your hemichordates down here, actually. So these are the groups that are often what we consider the outgroup to chordates. But you can see that the echinoderms are sister to, or just next to, right, the hemichordates. And so this is a really good tree that's based on a lot of really nice characters. You can trace them back nicely to common ancestors, right? Okay, so here's a classification of reptiles that is monophyletically sound. So notice here, down here is what we call our most recent common, or I should say our plesiomorphic outgroup, right? Here's our outgroup, which we're using here as mammals, notice. What I want you to notice is mammals have one of the longest lines of evolution here. So they're a very old group, and hence why they're very, very derived. But mammals look a lot different than the rest of the tetrapods here, or the amniotes, right? It's a better term, the amniotes, which includes birds are included in here, and all of reptiles. If you exclude birds from this monophyletic group, then it's paraphyletic, right? So birds are monophyletic to reptiles, at least in this scheme, right? So basically, I love this cartoon. It says it means that reptiles don't exist. And no, it means that you're one of us. So there's some arguments here. But in reality, there had to be some major reorganization of how we group reptiles. And I'll talk about that when we get to the reptiles, based on some new genetic information and also including birds, right? So aves used to be kind of its own class, but it's not anymore. It's actually part of what we call class Reptilia. Or there are some new classes, if you will, that have been devised over the last 5-6 years that we'll get to when we talk about this bird dilemma. Also, the location of turtles is heavily debated within reptiles, and so we'll kind of talk about that when we get to reptiles. Here's a great example with foxes, right? You might see pictures of foxes. And what I want you to notice here, these are all carnivores, right? You can see there's dogs and foxes and whatnot. But not all foxes are closely related, even though they look a lot alike. So if you look at these two, look at this guy up here. So here's the red fox that you find here in North America. And here's the gray fox that you find in Africa. And they look a lot alike as far as their morphology, a little bit different in color. You can get some red foxes that actually have some gray on them. And you might immediately say, my gosh, these are a lot different than dogs. These two must be closely related. And you can see on this tree that they're really distantly related, right? There's some foxes in here too that look more dog-like, right? You can see that they kind of fit in here. But all of the foxes of the world are not a monophyletic group. They're actually polyphyletic. These African foxes are obviously way far away or distant from the red foxes you find here in North America, and they can't interbreed, and they've been separated for a long time, right? So they are actually separate groups, and they evolved to look a lot alike because the environmental niche that they occupy in Africa is similar to the environmental niche and what they eat here in North America, and they look a lot alike, even though they are not closely related. So that's an example of polyphyly or polyphyletic. Okay, our last slide here is called abundance phylogenies. This is when you, so this last slide is called abundance phylogeny, and this is when you basically try to include the number of species, how speciosus a group is, on top of kind of how they're related. And often with these abundance phylogenies, they put the time scale, the geologic time scale up here so you can see when certain groups were very abundant and when they went extinct or when they are less speciosus or have less species. And you can see these are different groups that come across here. You can see some of these groups are really large and always have been. So this really wide group here that's very speciosus, has lots of groups, is called Osteichthyes, or the bony fish. And bony fish are a very, very dominant group. It's the most, they make up 50% of all vertebrates, bony fish do. You can see the reptile line here. And then the aves line coming off. And notice birds are much more abundant than reptiles now because they can fly, right? And having flight makes you very successful. So there are lots of different groups. The first group we're going to talk about here, agnathans, are very, very, very underrepresented in living species. They're not speciosus. They used to be way back in the day, all right, when vertebrates first arose. These are called jawless fish. They're round-mouthed fish. They have a round mouth with non-movable jaws. There are a couple species alive today, and that's all that's left over from this very diverse group that arose kind of during the early evolution of vertebrates and chordates, and we'll talk about those guys first. We're going to kind of move across here like this when we discuss our organisms. Now, there are two rates to evolution, or there are lots of different rates to evolution nowadays. But what I want you to realize is that evolution is real, as you already know, right? But the mode of it can be different. What I mean by that is Darwinian evolution is slow evolution, right? It's change over time that takes millions and millions of years. There are events, like you can see right here, where you go from nothing to lots of animals. That is called punctuated equilibrium. And this is when all of a sudden you get this explosion of organisms that show up. They typically happen after mass extinction events on the planet, where you open up all of these niches and then organisms go in to fill it. And they diversify into a bunch of species that may still be around today or might be extinct depending on when that event occurred. And that's called punctuated equilibrium when you get to this explosion of evolution. Of course, when Stephen Gould, who's now dead, came up with punctuated equilibrium and showed evidence for it, there were two schools of thought. There was gradualism or Darwinianism and punctuated equilibrium. But in reality, some groups undergo both of these, where you get this huge diversification and then stasis with really slow change, and huge diversification and stasis, and huge diversification and stasis. So they're combinations of these during the evolutionary line of many of the organisms as far as the fossil record goes. And it all depends on when niches open up and when niches are closed. It depends on how fast things can evolve versus how fast they can't on the macroscopic scale. All right, so that finishes up our introduction to evolution, cladistics, and some of the terminology we're going to need for lab. Now we can start jumping into the chordate groups and start discussing them. So that's what we're going to do next. I will start with the hemichordates. So look for that lecture here this week. Have a great day.